All right, so as I was saying, guys, so we worked with Chromosocks the other day. So what we're going to do today is we're going to run through a breakdown of how this lab goes. We're modeling all the phases of meiosis. All right, so we start off with our 3 pairs of socks. So we said yesterday what these socks represent. What were those?
Chromosomes.
Chromosomes, yes. Good job, both of y'all. All right, so these are representing our chromosomes. So this particular cell would have how many chromosomes?
6.
6 chromosomes, yes. How many homologous pairs? 3 homologous pairs. So we've got 3 copies of the same chromosomes. So if I've got 1 of each, is that going to make that, or if I've got 2 of each, is that going to make that haploid or diploid?
Diploid.
It is diploid, yes. All right. So this is what our cell looks like in interphase. We've just got our laundry basket of socks right here. They're all discombobulated, jumbled up mess. So at the end of interphase, all of these guys start coming together and kind of aligning a bit. They form a nice, neat pair. Nice and neat. All right, so we're going to enter meiosis 1. Meiosis 1 has 4 stages. PMAT. What's the P stand for?
Prophase.
Prophase. M.
Metaphase.
A.
Anaphase.
T.
Telophase.
Good job. All right, so we're going to go through prophase first. So in prophase, what's the first thing that all of our DNA does? That all of our chromosomes do?
Together? Like get
They're
with
together.
themselves?
Well, when you say get together, what do you mean?
Like get with their kind?
Get with their kind, join?
Like that gets with that.
Close, but not quite. We actually duplicate. So it's not that this chromosome joins with this one, but this one is going to duplicate. It's going to make another copy of itself. So we're going to model that using a rubber band and another pack of socks. So we're going to put these two guys together, rubber band them, and this rubber band is going to act as our centromere. Okay, the centromere is the part on the chromosome where it's duplicated. Okay, so at this spot, this chromatid is going to be created. So now we've got 1 chromosome with 2 sister chromatids. All right, so all of our DNA is going to do that. I'm going to pass these out and let y'all rubber band some of those together. All right. Okay, there's a green one for you. There's a black one for you right there. Here, do this green one right here, hon. All right, so go ahead and rubber band those together. Yes. You may. No, you may not. You can't right now. Ask me in 5 minutes.
It's gonna be gone in 5 minutes.
Well, it'll go in 5 minutes then. All right, so once you got them all replicated, go ahead and put them in the middle. All right, rubber band them and throw them in the middle of the table. Yep.
Why are your fingernails purple?
Nail polish.
Yep.
Neat.
Very good, very good. All right. Okay, so now we've got all of our DNA replicated. So we're going to pretend that our nuclear membrane has already broken down. Chromatin fibers, or not chromatin fibers, I'm so sorry. Spindle fibers are already forming, and they're going to grab onto these centromeres. Wait, yes, ma'am.
Bye.
She's on her way.
She's talking about
Bye, Addie.
Addie.
All right, so prophase is done. Now we're going to go into metaphase. What happens in metaphase? Bye, Addie.
One of season's not in
All right. Huh? Yes.
Okay, middle.
Okay.
Start December.
Okay, guys. Quiet. We're going to answer that one more time. Boys. All right. So my question was, what happens in metaphase?
The genes copy and they all line.
They will. They all line up in the where?
Middle.
In the middle. So they are all going to pair up and line up in the middle of the cell. All right. So looks a lot like this. All right. So now that they're all lined up in the middle of the cell, that's metaphase. Now we're going to go into anaphase. What's our keyword for anaphase?
A.
What's our keyword for that?
Oh, apart.
Apart.
No, not.
So they're going to be pulled apart. So what happens to pull these apart?
You gotta pop the ribbon.
Nope. What happens to pull these guys apart?
You just pull them apart.
Spindle fibers.
Yes, the spindle fibers start kind of contracting. The spindle fibers start pulling these guys to the other side. So guys, go ahead and separate these two into two different cells. All right, so now we've got our two cells. So Anaphase is done. They're all apart. And now we're going to enter Telophase. What happens in Telophase?
They come in.
Nucleus reforms, so you would have two. So you end up with two cells. So these guys break down the middle, you end up with two cells. So we're going to pretend this is two cells right now. But our story for meiosis is not over yet. In meiosis, it goes through this division twice. Now, in meiosis 1, we had to duplicate. Does that happen in meiosis 2?
No.
No. So we're going to do meiosis 2 one cell at a time. So we've got two cells now. So if we do meiosis 2 with this one, we don't duplicate, but what happens? Prophase, what's going to happen with this one? Prophase of meiosis 2. We'll prepare to divide, right?
Right.
Okay, so we're going to prepare to divide. Let's go ahead and enter metaphase. What happens in metaphase?
The genes copy and align down the middle.
Okay, do they copy in metaphase 2? In meiosis 2? Do the genes copy in meiosis 2?
No.
No. So they just line up down the middle again. So there's no copying. How many chromosomes do we have here?
6.
We only have 3. We've got 3 chromosomes.
There's 2 on each.
We've got 6 sister chromatids, but we've only got 3 chromosomes. All right, so now we got metaphase. We go into anaphase 2. What happens in anaphase 2?
They spread apart.
They spread apart. Okay, so in order to do that, what do I need to do?
Take the rubber band off.
Take the rubber band off. So y'all go ahead and model it with that one. We'll model it with this one.
Yeah.
All right,
Can you keep
so
'em?
take the rubber band apart, and then what happens?
They come together.
Not-
No, they go apart. Apart.
Apart.
Anaphase.
Okay. Anaphase apart. And then telophase. What happens in telophase?
Whoa.
They create a new nucleus.
Nucleus reforms. So let's go ahead and form 3 stacks of socks. 3 stacks of
Oh,
socks.
my bad.
All right. So now we've got 3 different cells. Okay, 3 different laundry baskets. Sorry, 4 different laundry baskets, 4 different cells. Are all of these cells exactly the same? Or do they all contain the same type of chromosome? Do they all contain the same types of chromosomes?
Yes. No.
Do they all have an orange, a green, and a black?
Yes.
Yes. So, do they compare this to the original cell? Do they have the same number of chromosomes?
No.
No. How many chromosomes does each cell have?
3.
3. So does that make this haploid or diploid?
Diploid.
Haploid.
Haploid. Haploid. Think haploid half. Okay. Haploid half. Yes, ma'am. Tyus is on his way.
I think they make that thing louder.
All right. Okay. Questions, comments, concerns? No. All right. So we're going to go ahead and pack these things back up, and y'all are going to do this lab tomorrow. You can go ahead.